Thank you for joining us today for our Pesticide Detectives program finale. We would like to begin with an acknowledgement of country. We acknowledge that Pesticide Detectives was conducted on land and in waters sacred to the traditional custodians of the land. I would also like to acknowledge the traditional owners of the land that I'm speaking from today, the Wurundjeri and Bunurong people, and I pay my respects to their elders, past and present, and to the ongoing living culture of Aboriginal and Torres Strait Islander peoples. My name is Kavitha. Thank you everyone for attending our program finale on 17th of November 2020. This is a re-recording of that presentation. I joined the AQUEST group in 2018 and I've been working on two citizen science and community-based projects called the Litter Trackers and Pesticide Detectives. Today I'm going to talk about Pesticide Detectives to conclude the project. I very much enjoy working with students and grassroots organizations, and I'm very interested in waterway health, and this was an immensely rewarding opportunity to work on the Pesticide Detectives project. Why pesticide detectives? Humans have made 50 million different chemicals here on Earth, the vast majority over the last few decades. Our regulatory bodies simply cannot keep up with the many new chemicals that are being developed every year. Most chemicals on the market have not been tested, and we don't know if these are ending up in the environment and if they are of concern. Why citizen science? Involving citizen scientists in this project enabled a nationwide snapshot of pesticides and provided an opportunity for people to participate in scientific research. With the data contributed by citizen scientists, we were able to see if there are patterns across Australia in terms of pesticides and if we need to direct further investigations. Some of you may not know about the project, so I'm going to briefly explain what was done. The Pesticide Detectives project started with participants nominating local waterways to collect sediment from. Participants then received over 77 sampling kits containing equipment such as glass jars, buckets, trowels, and consumables such as gloves. Our participants were involved with activities such as organizing and confirming collection dates, conducting risk assessments, recording information for each site, and taking and uploading photos. We thank our participants again for the amazing work and time that they had put into the project. Our participants collected sediment for us in jars, which we then received and sent to the laboratories for analysis. We chose sediment samples rather than water samples because sediment is relatively stable to transport in the post and can go for weeks without preservatives or chilling. Water samples cannot. Analyzing sediment gave us information on the types of pesticides that persist in the environment, and these types of pesticides would usually adsorb to the sediment quickly once they got into the water, rather than continuing to travel downstream with the water flow. The limitation of using sediment is that in the field, we usually filter sediment with a net to collect the silt or clay, also known as mud. However, this would be difficult for our participants to do. This meant that we received a whole range of sediment mixtures, including sandy sediment. Pesticides tend to adsorb better to mud than to bigger grain sizes such as gravel or coarse sand. In the end, the Pesticide Detectives project provided a new way for our participants to engage with their local waterways. Most hadn't collected sediment before, and this provided an opportunity to learn a new skill set and to find out what types of pesticides were present in their waterways. We tested for 110 different pesticides. This list is available on our website. It includes insecticides, fungicides, and herbicides. Insecticides also includes a class of pesticides known as synthetic pyrethroids, which is shown in green in the table. You may notice that glyphosate is not listed and wasn't tested for. This is because the method for testing glyphosate is different from many others. The pesticide screen we used has provided us with a wide range of pesticides. Alternatively, for similar money, we could have just tested for glyphosate. We decided to get the screen done as we were unsure of what would be found, and we didn't want to limit the project to focus primarily on glyphosate. We launched the project in April 2019 with participants from Waterwatch and Estuary Watch Victoria taking part in a pilot study of 13 sites around the Bellarine area to the Surf Coast in Victoria. For our pilot study, we tested for heavy metals in addition to pesticides. We didn't find any pesticides in our pilot study at the 13 sites, but we found 3 metals that exceeded the sediment quality guidelines at 5 sites. These were arsenic, nickel, and zinc. Heavy metal results are on our website. We had 48 groups and over 100 participants in our project. We had 21 groups in Victoria, making up the majority of participants. Being a home base, this is where we are more active. The last round of sediment samples were collected and concluded in July 2020. Participating groups, including some of you who are here with us today, are listed here. And along the way, we also had many other groups participate in this project. We are thankful for groups and participants that were able to join us and contribute their time and efforts to the project. 2020 summer was a particularly challenging time for our participants because of fires, floods, and COVID-19 impacts, and unfortunately, some participants were not able to complete collections, but we thank them also for taking the time to register and nominate sites. Here are some pictures of participants sampling around Victoria. The top picture is of us sampling around Colac in Victoria with Deirdre and participants from Estuary Watch. I also had the opportunity to join Peter Stevens and participants from Ozfish East Gippsland, Victoria to collect samples from the area. And the bottom right is a picture of Julia from Merri Creek Management Committee in Melbourne, who participated eagerly in the project with permits during COVID restrictions. These are pictures of Manjimup Senior High School in WA, who went out to collect samples as part of their science class, and this trip was organized by Andy Russell from Warren Ribbons of Blue. Sediment samples were collected from 240 sites across Australia by our participants. The number of sample sites were different across Australia, so it is difficult to make comparisons because of the uneven sample numbers. We had 83 sites from Victoria, which had the largest number of sites. Pesticides were detected in sediment from 32 out of the 240 sites, which is 13% of sites. This shows the breakdown of number of sites where pesticides were detected in each state and territory. These sites are surrounded by various types of land uses such as recreational, agricultural, viticulture, cropping, livestock, urban and rural residential, and industrial. You'll be able to have a good look at the surrounding areas around sites on our Google map on our website. If we look at the percentage of sites with pesticides, ACT would have the largest percentage of sites with pesticide detects. Again, it is hard to make a comparison because of the different number of sites across Australia, and sites were chosen by our volunteers based on different reasons, such as wanting to know about their local waterway health or choosing sites that were of concern. Our pesticide screen included 110 types of pesticides. The comprehensive list is on our website. Here's a list of pesticides that were detected in the sediment samples. 12 types of pesticides were detected. We found eldrin and deldrin, which are legacy organochlorine insecticides that were phased out in Australia together with other organochlorines in the 1990s. We didn't find any DDT, but we did find DDT breakdown products DDE and DDD. We found chlorpyrifos, which is an organophosphate insecticide used to control different kinds of insect pests. In Australia, these product registrations were cancelled in July 2020. In the EU, use of chlorpyrifos has been banned since the beginning of this year. This ban also includes food and fruit exports into the EU, bearing traces of chlorpyrifos. We found bifenthrin, fenvalerate, and permethrin, which are synthetic pyrethroid insecticides used in a combination of home gardens and agriculture. Synthetic pyrethroids were manufactured to replace pesticides that had been phased out, such as organochlorines. We also found fungicides tebuconazole, propiconazole, and iprodione, which are used in home gardens and agriculture. And we found diuron, which is a broad-spectrum herbicide and algicide used to control weeds in agriculture. Here is a list of the number of detects of each pesticide. Bifenthrin was the most common pesticide detected at 23 sites across Australia. We found dieldrin at 4 sites in New South Wales and DDE at 4 sites in New South Wales and South Australia. The rest of the pesticide detects were low at up to 2 detects. Looking at legacy pesticides, even though aldrin and dieldrin were phased out in Australia in the 1990s, these pesticides are still around in sediment, showing evidence of persistence in the aquatic environment. There is not much we can do to remove legacy pesticides. We just have to let them run their course. DDT is an organochlorine insecticide used to protect agricultural crops against insects in Australia and for mosquito control worldwide, and is banned in Australia since 1987. DDT was also manufactured for use as an insecticide. We found DDT breakdown product, DDE, at 4 sites. Concentrations at 2 of the sites, Bakers Creek in Victoria and Onkaparinga River tributary in South Australia, exceeded default guideline values. Concentrations at the other 2 sites in Wee-Waa and Narrabri in New South Wales were above the upper guideline value, suggesting possible impacts to aquatic biota. Bifenthrin was the most common pesticide detected at 23 out of 240 sites. These sites were mostly urban with established neighborhoods. 2 of the sites were new urban developments. The rest of the sites were mixed land use types or sites around crops. Toxicology tests for bifenthrin were conducted by Yepe et al. In 2017 in our laboratory on freshwater crustaceans, which showed that concentrations at a certain threshold pose a risk to freshwater crustaceans. Concentrations exceeded this impact threshold at 8 sites. The highest concentrations of bifenthrin were found in sediment from Orphan School Creek in New South Wales and from the Darling River in New South Wales. Orphan School Creek was retested because previous levels of bifenthrin also exceeded the threshold. The other 5 sites with concentrations exceeding impact thresholds were urban sites, and 1 site was around sugarcane crops. We can say that where threshold values have been exceeded, toxicological impacts on aquatic biota are likely. What does this mean for your site? Further investigations are necessary to find out if these sites show high concentrations repeatedly. It is also necessary to conduct more studies to find out specific impacts in the laboratory and in the field on aquatic biota before we can make any conclusions about the impact on ecosystems. So, a little bit of information about bifenthrin, as it is the most detected pesticide in our project. We are using bifenthrin as a case study to show how we can make change in this space. This work was conducted in Victoria by our research group, and we think the findings are relevant for everywhere in Australia. Pyrethrins are a natural form of pesticide found in chrysanthemum flowers. Bifenthrin, permethrin, and fenvalerate are a class of synthetic chemicals that are structurally similar to and mimic the action of pyrethrins. Synthetic pyrethroids are found in many household insecticides in Australia and are used on pets, wool carpets, on clothing, for mosquito control, and in agriculture. They are also used in new housing estates for termite control. These pesticides are not authorized for use in the EU. They are persistent and insoluble and accumulate in sediments or suspended particles than in water, which is why it is important to include sediment for analysis. They are not so toxic to mammals, but highly toxic to fish, crustaceans, and other animals. These pesticides have effects that we are only finding out about now, and which we will continue to find out about more in the future. In wetland studies around Melbourne, synthetic pyrethroid pollution is increasing in prevalence. In 2011, 33% of urban wetlands tested contained pyrethroids. In 2016, this percentage had grown to 86%. They are more commonly found in urban areas rather than rural areas of Victoria. Bifenthrin is used for termite control in new housing developments. Their use is also increasing in rural areas. From this wetland study, it was obvious that synthetic pyrethroids are an increasing issue, and further investigation ensued. Water-sensitive urban design treatments are primarily wetlands that treat urban runoff, and it has been found that synthetic pyrethroids remain in the waterway after passing through these treatments. So a study to further investigate the sources of bifenthrin was set up. We needed to confirm whether termite control is a major source of synthetic pyrethroid pollution, to determine how these insecticides get transported into local waterways, and to determine whether current labeled use and associated approved practices need to be reviewed to reduce synthetic pyrethroid pollution in waterways. We designed a study to investigate 4 housing estates across Melbourne. We looked at housing estates over time and selected properties from a vacant block to full completion. What we found was that widespread contamination of sediments with synthetic pyrethroids was occurring in newly constructed estates. The majority of houses treated with synthetic pyrethroids did not cause substantial pollution. A small number of houses post-construction caused substantial pollution. Professional application of pesticides did not cause this pollution. Surface runoff from buildings did not appear to be a major cause of contamination in the estates. As runoff was not a major source of dispersal for bifenthrin, we wondered if airborne dust could be a factor in the dispersal of bifenthrin. We tested dust around the new housing estates, in gutters, on power boxes, and on playgrounds, and found that synthetic pyrethroids were present in the dust. This study found that not just stormwater runoff, but also dust is important in the movement of bifenthrin, and by extension, other synthetic pyrethroids in the environment. Dust can move these contaminants long distances from their original application. There are a number of management actions available which require collaboration to proceed, including better education about the presence and use of pesticides, soil management, and potentially changes to council bylaws. Conversations with all stakeholders are really important to be able to work through solutions to the problem of pesticides in our waterways. A report has been prepared for government, and stakeholders have been engaged in an ongoing discussion. So watch this space. This was a question asked by a number of our participants who thought that there would have been pesticides detected at their sites, but were surprised to find none. Why is this so? Pesticides that we are screening for may not have been present in the sediment at the site, which is good news. New pesticides are being created every day. While we have screened for an extensive list of pesticides, there may not be a test available to detect other pesticides that may be present. Pesticides that could have been present may not be in our list of pesticides B that we tested for. Some pesticides are more water-soluble and, if present, may not have adsorbed to sediment sufficiently for detection to occur. Dynamics of the waterway may mean that pesticides could have been present in sediment in different locations of the waterway, but not at the specific sites that were sampled. Quality of sediment that was collected may affect detection of pesticides. Pesticides may not adsorb to sandy or coarse sediment compared to mud. We have come to the end of our talk, and to summarize some important points, we can confirm that Bifenthrin is present Australia-wide and not just in Victoria, as found in some of our earlier studies. The AQUEST group continues to conduct research on Bifenthrin. Legacy pesticides are still being detected in sediment 30 years after they were banned, showing their persistence. Types of pesticides found in relation to specific land use is not very clear. We may spend a bit more time later exploring land use around sites where pesticides were detected. We also learned that partners are crucial for citizen science project success. So we thank you again for participating and helping us with collections and promoting the project. We found that this citizen science project increased public awareness of pesticide issues and interest in the health of waterways where participants are from. More importantly, citizen science is a fantastic way to expand the reach of projects into locations that may not ordinarily be included. So where to from here? While Pesticide Detectives as a program is now complete and we don't have further funding to continue, if you are concerned about your local waterway, then it would be a good idea to get in touch with your local water authority. And we are always happy to speak to any organization or individual requiring information. We are likely to have future citizen science projects, and this could have a different focus, for example, microplastics or litter. So if you're interested in our future projects, we will put up a link on our website shortly to join our mailing list so that we can keep you informed. For a full list of results, go to our website at www.pesticidedetectives.com.au. I would like to acknowledge the contributions by the rest of the team, Dr. Jackie Myers, Professor Vincent Pettigrove, and Monica Tuman, for the project success. Thank you for your attention.